So membranes encase every cell and are made up by a fluid mosaic model. And this means that the membrane is composed of mostly phospholipids, cholesterol, and proteins, and that all of these move freely. So I'll start with phospholipids, which are lipids made of glycerol, 2 fatty acid tails, and a phosphate-linked head group. The phospholipids are these brown circles with the tails. And then the two layers, here's one layer and then here's another layer, make up a bilayer, which is a stable sheet-like structure. And the heads of these phospholipids are hydrophilic. The heads are the circle, all of these. These are the heads on the outside of the membrane. They're hydrophilic, which means they're water-loving, and the heads are on the outside of the membrane, like I said. And the fatty acid tails are hydrophobic, meaning water-fearing. These are the tails are the two coming off of each head inside the membrane. And the two nonpolar fatty acids extend in one direction parallel to each other. Obviously, they're inside the membrane. And the other polar phosphate group points in the other direction. The fatty acid tails determine the fluidity of the membrane. So for example, glycerol phospholipids that are saturated tend to make the membrane less fluid because they pack together well. And in the membrane, phospholipids can move in different ways. One way is laterally, which means the phospholipid moves around in one leaflet. So basically that would mean the phospholipids move in one part of the bilayer, and the other movement is a flip-flop movement where the phospholipids move between both leaflets of the bilayer in a transverse movement. And the transverse movement allows asymmetry to be maintained in the membrane, so that would mean that they would switch places, basically. The next part that makes up a membrane are proteins, and proteins are the second major component of the membrane. And there are two different types, integral and peripheral. Integral proteins can integrate into the protein, which means that they can move, that they go all the way through the protein. So these are examples of integral proteins. They have at least one hydrophobic region that anchors them to the hydrophobic core of the phospholipid bilayer. So this is the hydrophobic core. And some integral proteins go partially into the membrane while others go all the way through, and those are called transmembrane proteins. So the two I just pointed to are both transmembrane proteins. And some integral proteins form a channel which allows ions and other molecules to pass through. That's an example of that, where a channel. And the stretch of the integral protein within the hydrophobic interior of the bilayer is also hydrophobic, made up of nonpolar amino acids. And like the lipid bilayer, the exposed ends of the integral protein are hydrophilic, so the outer parts are going to be hydrophilic, while the inner part of the protein is going to be hydrophobic to match the inside of the membrane, which is also hydrophobic. And integral proteins don't flip-flop, but they can move laterally in the membrane. And peripheral proteins are the other type, which are found on both the inside and outside of the membrane. And they are usually attached to phospholipids or integral proteins, but they can't cross the membrane. And they don't interact with the hydrophobic core of the phospholipid bilayer. Instead, they're usually bound to the membrane indirectly by interactions with integral membrane proteins or directly by interactions with lipid polar head groups. And peripheral proteins can move laterally but not transversely because they don't go through the membrane. Here's an example of a peripheral protein sitting on this protein outside of the membrane. The next thing that makes up the membrane are sterols, and sterols such as cholesterol are important to reduce membrane fluidity and permeability, and they help to increase membrane rigidity and strength. And sterols consist of a set of fused carbon rings. 3 with 6 carbons, and 1 with 5, and the rings are decorated with other groups. So for example, hydroxyl groups, which are hydrophobic. Here's an example of cholesterol inside. And sterols are hydrophobic and are found among the hydrophobic tails in the lipid bilayer. So here, they're with the hydrophobic tails. And sterols don't move independently, but if the phospholipid they are attached to moves, they would move with that phospholipid. The next thing that makes up membrane is carbohydrates, and membrane carbohydrates are chemically bound to glycolipids and glycoproteins. That's here. That's a glycoprotein, and this is the carb attached to it. And most carbohydrates are found on the outside of the cell, however they can be found on the inside sometimes. And carbs are generally hydrophilic, as most of them reside on the outside of the cell, which is water-loving. And carbs don't move laterally independently like the sterol, but if the protein that they are binded to moves, they would move with it. So yeah, that's the end.